Good morning, everyone. My name is Davide Croci, and I'm a biomarker scientist at Novigenics, a precision medicine company specialized in AI-driven RNA analytics and immune response profiling. Today, together with my colleagues Sandra van Wilpe and Niven Mehra, we will present our recent study published in JCI Insight.
Hello, my name is Sandra van Wilpe. I'm a postdoctoral researcher and a medical doctor at the Radboud University Medical Center.
And I'm Niven Mehra. Good morning, everyone. I also work at the Radboud University Medical Center as a medical oncologist and associate professor. And as many of you know, immune checkpoint inhibitors, a type of immunotherapy, have significantly improved survival in patients with metastatic urothelial cancer. However, only about 1/3 of the patients respond to immune checkpoint inhibitors in monotherapy, and this highlights an urgent need for more biomarkers that can distinguish between patients who will benefit and those who don't. Our study aimed to identify biomarkers for the early detection of patients without clinical benefit from treatment, in order to avoid unnecessary exposure to ineffective therapies and to enable an early switch to other therapies.
To achieve this, we focus on detecting tumor-derived signals in the blood. Why blood? First, blood collection is minimally invasive and can be done repeatedly over time, making it ideal for patient monitoring and clinical implementation. Second, the blood is indeed a rich source of tumor-derived signals. To measure these signals, we use two complementary technologies. First, we analyze gene expression changes of immune cells in whole blood samples, what we call the immunotranscriptome. And second, we study circulating tumor DNA, ctDNA, which is released into the bloodstream by tumor cells and serves as a direct indicator of tumor burden. By integrating these two approaches, we obtained a 360-degree view of the blood-derived signals, capturing both immune and tumor-derived changes.
So in our study, we analyzed ctDNA and whole blood immune transcriptome data from 93 patients with metastatic urothelial cancer. Patients were treated with checkpoint inhibitors as first-line or second-line therapy, or as maintenance checkpoint inhibition after a favorable response to chemotherapy. Blood samples were collected at baseline and after 2 to 6 weeks of treatment. And clinical benefit was defined as progression-free survival beyond 6 months. So in 88 patients, we performed ctDNA analysis. We categorized patients into 2 groups based on the ctDNA dynamics. Patients with an early increase in ctDNA were predicted to have no clinical benefit, while patients with stable or decreasing ctDNA levels were considered likely to respond to therapy. Our ctDNA-based approach demonstrated high specificity of 92%, meaning that nearly all patients who benefited from treatment were correctly classified. However, sensitivity was only moderate at 59%, meaning that we could not identify all patients without clinical benefit.
At this point, we wanted to evaluate whether integrating whole blood immune transcriptome data could enhance our predictions. Using Novigenics' proprietary LithoSeq platform, we analyzed patient samples, and after quality control and RNA analysis, we identify key biological pathways associated with immunotherapy response, including T cell activation, interferon signaling and antigen processing, and cell cycle regulation. These pathways have been previously linked to immune checkpoint inhibitor response, but we also discovered novel biomarkers, such as non-coding RNAs. Based on these insights, we then developed a 10-gene predictive model for immunotherapy response. To ensure model reliability, we divided our cohort into 3 subgroups: a discovery cohort for model training, a test cohort for initial model confirmation, and a blinded validation cohort for independent assessment. As shown in the graph, specificity and sensitivity, our parameters to evaluate model performance, remain high and stable across all cohorts, indicating a robust ability to predict patient outcomes. Next, we explored whether combining ctDNA and RNA-based biomarkers could further improve predictive performance. In the plot on the left, we compare sensitivity and specificity across different models. The ctDNA-only model in orange, the RNA-based model in green, and the combined multimodal model in violet, shown for both test and validation cohorts. In the validation cohort, the multimodal test achieved an outstanding 100% specificity and 79 sensitivity, significantly outperforming both standalone approaches. And as seen in the graph on the right, the multimodal model effectively distinguishing between patients with and without clinical benefit.
So to summarize, our biomarker approach enables us to identify 40 to 50% of patients without clinical benefit. And by using this test, checkpoint inhibitors in these non-responders may be discontinued early so that these patients can be redirected to alternative therapies. So by enriching for patients that are likely to benefit from treatment, our test would lead to increased response rates in those patients who continue checkpoint inhibitors. And we are currently validating these promising results in a new cohort of patients. So before we conclude, we would like to express our gratitude to the patients and their families whose participation made this research possible. And we also want to thank our collaborators and our funding partners for their invaluable contributions.